Now we will talk about chemical digestion. Starts with the mouse taking food that is ingested. The mouse, as I've been told before, only chemical digestion of cooked starch by the enzyme amylase, which is a thiolene. The mouse leads to the buccal cavity, to the throat or pharynx, followed by the esophagus. The food is forced down by the gullet by peristalsis to the stomach. The stomach has a strong muscle walls, as we said before. It's called the pyloric sphincter muscle, which is a circular band of muscle at lower end of the stomach that stops solid pieces of food from passing through the duodenum. The main function of the stomach is actually three functions: store food, turn food into liquid, and release food into small quantities at a time to the rest of the alimentary canal. This peristaltic action of the muscles in the wall of intestine is a kind of a physical digestion. These muscles contract and relax, squeezing food and mixing it with gastric juice, turning the mixture into a creamy liquid called chyme. So it starts from the mouth, called the bolus, and then it's transferred to another structure which is called chyme. So now the chyme will move to the small intestine. Continuing talking about the stomach contains different types of cells. And these cells situated in a pit, like the figure. This is actually a gastric pit in the figure. This gastric pit in the stomach wall which actually secrete the gastric juice. These pit contain important type of cells called goblet cell, as you can see from the figure. These goblet cells secrete the mucus. Other cells which produce the main protease enzyme, which is the pepsin, other make hydrochloric acid. The pepsin, of course, digests proteins into polypeptide, and this pepsin must work in acidic condition. In renin, what's renin? Renin is only produced in the stomach of young mammals to clot milk they got from mother. So the milk protein are then broken down by pepsin. After 1 or 2 hours, the sphincter at the bottom of the stomach opens and let the chyme pass into the duodenum. And you can see from the figure the cells which will produce mucus, then followed by cells which makes pepsin, followed by cells which make hydrochloric acid. And these constitute the cells of the gastric pit. In this figure, you can see the relationship between stomach, liver, and pancreas. You can see where is gallbladder is located, where is the pyloric sphincter is located, and then the stomach is followed by the duodenum. The hydrochloric acid secreted by cells in the wall of the stomach creates a very acidic medium, reaches a pH 2. This pH is important because of two factors. It denatures enzymes in harmful microorganisms in food, bacteria, so bacteria will die in the stomach due to acidity, and as well it provides optimum pH for the pepsin to work. Let's talk about the small intestine, which are around 5 meters long. The pancreatic juice and bile from liver are produced into duodenum. The pancreas, which is a digestive gland lying below the stomach, it makes a number of enzymes which act on all classes of food. The protease, the pancreatic amylase, which is different from the amylase which is in the mouse, the lipase. The protease here is called trypsin, which breaks the protein into polypeptides and followed by amino acids, while the pancreatic amylase, of course, break down starch into maltose, while the lipase break down fats, lipids into fatty acid and glycerol. This pancreatic juice contains as well sodium hydrogen carbonate, which is particularly important in neutralizing the acid coming from the stomach. And this is necessarily because the enzyme working there in the small intestine must work in alkaline medium and do not work well in acidic condition. All the digested material now is changed into soluble compound, which can pass through the lining of intestine and into the bloodstream. If we were talking about the small intestine, we need to know the structure of the villi. The inner wall of all parts of small intestine, the abduodenum and the ileum, is covered with millions of villi. Each villi is about 0.5 to 1 millimeter long. Cells covering villi make enzymes. The enzymes do not come out into the lumen of the small intestine but stay close to the cell which make them. This enzyme will complete the digestion of food. The carbohydrate enzyme maltase breaks maltose into glucose. The proteases finish breaking down any peptides into amino acids. Lipase complete the breakdown of fats into fatty acid and glycerol. So the outer membrane of each villi cells have another structure for increasing the surface area, which is called microvilli, which increases the surface area by 20 times the exposed surface to the cell. So each microvilli have on its border, each villi have on its border another structure called the microvilli to increase the surface area 20 times. What is the significance of chemical digestion? Solid food is dissolved to make a solution. This breaking down of large molecule into small molecule to enable it to be easily absorbed. The small molecule can be absorbed through the epithelium of the alimentary canal, through the walls of the blood vessel and into the blood. So amylase in mouse secreted by the salivary gland, while in duodenum amylase come the pancreas. The proteases in stomach it's a pepsin, and in duodenum is trypsin, which is secreted as well by the pancreas, while the lipase in duodenum lipase secreted by pancreas. Starch is digested in two places in alimentary canal, by salivary gland by salivary amylase in the mouse, and by pancreatic amylase in the duodenum. This chemical breakdown of starch takes place in two stages. Amylase work best in neutral or slightly alkaline pH and converts large insoluble starch into smaller one, soluble 12-carbon maltose chain. The maltose is a disaccharide and too big to be absorbed through the wall of intestine. That's why maltose is broken down to glucose by maltase, which is present in the membrane of the epithelial cells of the villi of the small intestine. In this figure, we'll summarize the processes that's already happening. So if you think, the first figure, if you think that this is a large molecule, example starch, it is broken down by enzyme A, which is the amylase. It breaks down into peptides, which is the maltose. This maltose is attacked by a different enzyme and broken down into a single amino acids. The digestion of proteins. There are two major proteases break down proteins, as we said before, pepsin and trypsin. The pepsin is secreted by the stomach, while the trypsin is secreted by the pancreas. The pepsin convert the proteins breakdown into small peptides, shorter chains of amino acid, while the trypsin have a similar effect and have a similar role to pepsin, but works in the but secreted in the pancreas in the small intestine. The small intestine itself doesn't produce enzyme. We have crepes in the villi, so the villi is not a digestive gland, but can produce mucus. We have crepes in the villi. These crepes, the main function of rest is actually to renewing the cells that rupture due to the movement of food. So they produce new epithelial cells to replace that lost from the tips of villi due to the movement of the food. The epithelial cells of the villi contain enzymes in their membrane that complete the breakdown of sugars and peptides before they pass to bloodstream. So we have peptidase that breaks down polypeptides and the peptides into amino acids. Bile and its function. The bile actually is a yellowish-green fluid made in the liver, bile pigments. Hemoglobin from RBCs are broken down and pigments are excreted in feces. The bile is stored in gallbladder and delivered to the duodenum by the bile duct. Actually, the bile doesn't contain any enzymes. The bile is alkaline due to the presence of sodium hydrogen carbonate, which neutralizes the acidic mixture comes from the stomach and the gastric juice as it enters the duodenum. Enzymes secreted in the duodenum needs alkaline medium, so that's why there is a sodium hydrogen carbonate for optimum rate of reaction. The bile contains as well bile salts, which act on fat like a detergent for emulsification. So this emulsification is a kind of mechanical digestion. They break them into small droplets with a large surface area to be more efficiently digested by lipase to fatty acids and glycerol.